وعليكم ورحمة الله وبركاته. My name is Dr. Mohammed Hamdi. I am a professor of clinical hematology and internal medicine at Ain Shams University, Cairo, Egypt. Today's lecture is intended for undergraduate students who are taking the course of hematology. The aim of this lecture is to study the composition of the blood and the main investigations that will be needed in the course of the undergraduate for clinical hematology. Objectives for this course are understanding blood composition, to understand that the blood composed of plasma and packed cell volume or cells, and buffy coat or other cells, and key proteins. And to interpret the RBC's indices, we will understand what is meant by indices, the platelet function, and white blood cell differentials. And to master of diagnostic tools like blood film, electrophoresis, either protein or hemoglobin, Coombs test, and bone marrow analysis and blood grouping. And link this physiology to clinical scenarios like anemia, leukemias, hemolysis, and bleeding, etc. How we study blood. We introduce the needle inside the vein into this tube, which contains a purple-shaped head cap. We take about 3 centimeters of the blood, 3 cubic centimeters or 3 milliliters, and put it into this machine called centrifugation machine that will separate the blood into 3 parts. The first part, the clear fluidy part, which is called plasma, the opaque part, which contains the cells called the packed cell volume or the formed elements, and a semi-transparent part called buffy coat, which will contain some plasma and a lot of platelets and white blood cells. Now will compose about 55% of the whole blood that I have drawn from the vein, and 45% of that is the hematocrit or the packed cell volume or the formed elements, which contains the cells, and less than 1% of this amount is composed of this buffy coat, which contains again some of the plasma and some of the white cells and platelets, which is separated. This separation by the centrifugation will make the lighter things above, which is the plasma, contains only proteins, and the heavier things like the RBCs in the lower part, and the intermediate sizes and the intermediate shapes are present in the in-between, which is called the buffy coat. Contains a lot of things, mainly water, ions, proteins, gases, waste products, and nutrients. The bottom layer, the packed cell volume, contains exclusively the RBCs, which contains the hemoglobin, which helps in transportation of oxygen to the cells and taking carbon dioxide back to the lungs. And in between there is this small amount of elements which are called the buffy coat, which contains white blood cells and platelets. Composes about 55%, and the formed elements composes about 55% for the plasma and 45% for the formed elements. The whole amount of the blood in the body ranging between 4 to 5 liters in females and 5 to 6 liters in males as a mean. And it maintains a pH between 735 and 745 for optimal enzymatic activity that present in the plasma and for the function of all functions of the blood. More details of the plasma. The plasma composed mainly of water, 90%, and about 7% of protein and 3% of other soluble like electrolytes, like gases, like some nutrients and some waste products. But the most important composition of the plasma is the water and proteins. And some of these proteins are important proteins that carries more sophisticated things like lipids and like heavy metals. So it is important to know that the composition of the plasma also plays a role in the hematology, because some derangement of the plasma will lead to serious hematological diseases. The cellular component composed of mostly of the packed RBCs, which contains exclusively the RBCs. the packed cell volume, which exclusively contains the RBCs. It occupies about 38% to 48% for males, and a little bit lower for females, 35% to 45%. The importance of this numbers and this percentage is that it has some clinical relevance. If the packed cell volume is low, this means low amount of RBCs, and this means that the patient has anemia or overhydration, a lot of water, so the packed cell volume occupies less than the 45%. And if it is very high, this means that there's a lot of RBCs. This means the patient suffers from polycythemia, increase in amount of the RBCs or hemoglobin. Or sometimes it is a reflection of dehydration at that time. This is not true increase in packed cell volume. The buffy coat represents less than 1%. On the right tube, it is less than 1%, just like a film over the packed cell volume. But on the left tube, there is very increase in the buffy coat. This means that there must be increase either in the platelets or either in the white blood cells. This means the patient may have leukocytosis or leukemias. In detail, the function of every part of the plasma and every part of the formed elements, and the site of production and the major function of them. It is not important now to understand and rehearse all of this. But as we have discussed before in physiology, in the primary medical study, these things has passed for you in details before. But we will not need all of these in details in the next course, but just understand that the plasma and the formed elements composed of different things which has different site of production and different major functions. I will give you some hint for this. For example, some of the coagulation proteins are produced in the liver. So if this coagulation proteins like fibrinogen is not sensitized by the liver, the patient will suffer from bleeding. So it is important to know that even things that is produced in the plasma may have an impact on the diseases that we will discuss in the course. The plasma composition. Said before, plasma composed mainly by 90% of water and 7% of proteins. The proteins are here. Most of these proteins are albumin, 3.5 to 5.5 grams per deciliter, and maintains oncotic pressure and transporter hormones and the drugs. The globulins are the next major proteins in the plasma, which are composed of 35% of the whole components of the proteins in the plasma. And there are three types: alpha, beta, and the gamma globulins, each of which has function. The most important for us is the gamma globulins, because gamma globulins are the classes of the immunoglobulins, and immuno means it plays a role in the immunity. There are different kinds of immunoglobulins, but all of them are called gamma globulins because they are in an area of gamma globulins, specific shapes and a specific site and a specific size. Still, alpha and beta globulins are very important, even in hematology and in other diseases. Fibrinogen, it's about 4%, and it is a precursor of fibrin in clot formation, and it is important because it is one of the factors of the coagulation. Deficiency of fibrinogen will lead to bleeding. Other regulatory proteins like enzymes, proenzymes, and hormones, some of them are important for us in hematology, and some are important in other parts of the internal medicine and in medicine as general. The clinical importance of this composition to understand it that, for example, if the albumin is low by a liver disease, the patient cannot maintain the oncotic pressure and hence starts to have edema everywhere because it cannot take the fluids out from the extracellular fluid back to the plasma. So the extracellular fluid will be filled with free fluid, and hence the patient will suffer from edema. And for example, if the globulins is elevated, this means that gamma globulins is elevated. This means the patient may have infections, and sometimes he may have some kind of cancer called multiple myeloma. We will discuss this in details in the course. The kinds of important proteins in the blood, in the plasma, are the coagulation cascade. It is composed of multiple proenzymatic activity that will activate the second protein, the second enzyme, to reach the last step that form fibrinogen into fibrin that will cause fibrin clot and will make adhesion and stoppage of the bleeding because it makes a seal of the bleeding. It is divided into what's called pathways, the extrinsic pathway, which depends on factor 7, the intrinsic pathway that depends of serial activation of factor 12 to activation to factor 11, then activation to factor 9, then activation to factor 8, and what's called common pathway that both extrinsic and intrinsic pathways are sharing, that will activate factor 10 into active factor 10 with the activity of the active factor 5 to active factor 5, which will lead to activation of prothrombin into thrombin, and the thrombin is responsible for activation of fibrinogen into fibrin. Fibrinogen is factor 1 and the thrombin are factor 2. The extrinsic pathways we can measure the activity of extrinsic pathway, that is to say factor 7 activity, by a test called BT or prothrombin time. For the intrinsic pathway, another test to study all of these enzymatic activity and all of these factors are called ABTT or activated partial thromboplastin time. And the common pathway are measured by the measuring of fibrinogen. Activity of common pathway is measured by the activity of fibrinogen level and fibrinogen function, as well as extrinsic and intrinsic pathway tests, that is to say BT and BTT, also give a hint about the activity of the common pathway. The packed cell volume composed about 99%, and it is RBCs exclusively. We will discuss it later. But now we will discuss the buffy coat, which composed only of less than 1%. The platelets count in the blood represent about 150 to 450. This is range. If less than 150, it is called thrombocytopenia. If it is more than 450, it is called thrombocytosis. The white blood cells composed of 4 to 11,000 per cubic liters. If it is, and it's composed of a lot of cells. The most important of them are called neutrophils, and the second most important and second most common is the lymphocytes. What is the clinical relevance of understanding that the buffy coat? We understand that now that if the buffy coat is too much, like this case mentioned by the arrow number 1, this means there is a lot of white blood cells, and this can be interpreted if we make the blood film on the right panel of the picture. We will see a lot of large cells that compose of large nucleus. Some of them have nucleolus and open chromatin, and in between them there is a scarce and less of RBCs. The RBCs must be very abundant and just 1 or 2 of these white blood cells present in the film. But when we see a lot of white blood cells like this in the film associated with increase in the buffy coat, this means that there are a lot of white blood cells, and this means the patient has increase in the white blood cells. These cells may be normal cells with normal activity, and the patient has serious infection, and at that time it's called leukocytosis due to infection. But if the cells are very large like this, and it is composed of very early cells, then the patient at that time will be called having leukemia. Discuss the packed cell volume. The packed cell volume is composed of about 99% of RBCs, and the RBCs is rounded in nuclear cells, that is to say it has no nucleus, and composed mainly of hemoglobin and some fluids and some enzymes and some proteins, etc. To describe the RBCs, we have many descriptions. We describe it by its shape, we describe it by its size, we describe it by the amount of hemoglobin inside it, and we describe it by the concentration of this hemoglobin within the cells. All of this description are put together in a matrix, and they are called the RBCs indices, a plural of index. So each one of these is an index for description of RBCs by a mean. So the first index is called mean corpuscular volume. And mean, we means that the mean, the average size, and the volume means the average volume of all RBCs. The RBCs count in the peripheral blood is about 5 millions per cubic millimeter. So how we make the mean of single RBCs? We make this hematocrit, this means 45%, divided by the RBCs count. So it shows that about 45% of the blood is mainly packed RBCs. So we assume this is the mean packed cell volume. So the mean packed cell volume is 45%, and we call it hematocrit. If we divide it by the number of the single RBCs, it will give me the mean volume of a single RBCs. So the question comes here, why we consider the hematocrit is a whole volume? Because it's actually a whole volume. So we consider that we take 100 milliliter of blood, for example. If we make a hematocrit 45%, this means that it is 45 milliliter. So now we agreed that we have a 45 milliliter of hematocrit. Not just a percent, it is now an actual milliliter. So divided by the amount of the RBCs, which is 5 millions per milliliters. And here we come to the equation of the mean corpuscular volume and give me what's called the mean corpuscular volume. The mean corpuscular hemoglobin is the same. We take the 100 milliliter and we know that the most mean of the RBCs hemoglobin in the human is about 15 grams. So we assume that the 15 grams is the mean hemoglobin. We'll divide it by the count of the RBCs. We can say that what are the amount of hemoglobin in each RBC. Then comes the last one, which is the mean corpuscular hemoglobin concentration. How much this hemoglobin occupy within the RBCs? Is it occupying 90% of the RBCs? Does it occupy 70%, 50%, 30%, or 10%? This is very easy. We will divide the amount of hemoglobin over the amount of hematocrit. So we have, for example, 15 grams, and we have about 45 milliliters of the blood are RBCs, and the others are plasma and the papicoat. So at that time, we can understand how much this hemoglobin occupies of the RBCs. The last one is the RDW. It is something which shows the distribution of the mean corpuscular volume, the distribution of the volume over the curve. So it is just mere another explanation of the mean corpuscular volume. So it is just the distribution of this mean corpuscular volume. It is not independent form of measures. It is dependent on the mean corpuscular volume and its distribution. We will not discuss this in today's lecture, but we will use it in some of the lectures of anemias. For the RDW, we will not discuss now. So the normal hemoglobin level is between 13 and 17.5 for the males, and between 12 and 15.5 grams per deciliter for the females, and the average of them is 15. And for the newborn, the blood is much higher. It's about 16 to 20, and sometimes in some literature 22. So the mean corpuscular volume is 80 to 100 femtoliter. How it comes? We have this hematocrit, which is about 45. The RBCs count is 5 millions multiplied by 10 to measure it to the quadrillions of liters. So at that time, we will reach the number, which is about 80 to about 90. And it is a range about 10% above and below. So the mean corpuscular volume will be 80 to 100 femtoliters. And the femtoliter is the 10 to the powers minus 15 of a liter. Or from another way of discussion, it is 1 quadrillionth of a liter. So the mean corpuscular volume occupies about 100 of quadrillionths of a liter. If it is low, this means less than 80, the patient will suffer from, and the patient hemoglobin is low, the patient will have microcytic. The cells is small, so it is microcytic, and because hemoglobin is low, it is anemia. It's called microcytic anemia. The most common cause of it is iron deficiency. If it is high, that is to say the cells is larger than 100, it is called macrocytic, and if it is associated with low hemoglobin, it's called macrocytic anemia. As that kind, and it is caused mainly by B12 and folate deficiency are the most important etiology of it. Take care that sometimes the cells is small, for example, microcytic, but the hemoglobin is normal, for example, 14, and it is at that time it is not called microcytic anemia, it is just called microcytosis, and the same also can be explaining if the cells is larger than 100 femtoliter, but there is no anemia. It is now, at that time it is called macrocytosis, but without anemia. The mean corpuscular hemoglobin is between 27 to 31, and the mean corpuscular hemoglobin concentration is between 32 to 36%. About a third of the cell is occupied by the hemoglobin. If it is low for the mean corpuscular hemoglobin and mean corpuscular concentration, it is called hypochromia, and if it is high, it is called spherocytosis or hereditary disorders. At that time, the mean corpuscular hemoglobin concentration will be high. We will discuss this with the spherocytosis. The RDW is a measurement of the variation in the size, so it is dependent on the mean corpuscular volume. We will not discuss it separately. The platelets are small, discoid-shaped, and nucleated RBCs. We can see how much the platelets is small in comparing in this black arrows in comparison to the RBCs. And the white blood cells are even larger than the RBCs. So the platelets are very tiny. In the unactivated form, it is very flat like this, and in the left panel of the picture, in the upper picture, very flat and doesn't have any projection. But when it is activated, it will show some conformational change in the shape of the platelets, and it will show what's called pseudopedia, like false legs. It makes it more and more capable of adhering to other platelets and other proteins and causing the function of or intended function of the platelets. Its function is for primary homeostasis, to initiate clot formation, and to release growth factor for activation of other process of activation of the thrombosis. The normal count, as we have discussed before, between 150 to 450, and if it is low, the patient will suffer from thrombocytopenia and bleeding, and if it is high, the patient will suffer from thrombocytosis and risk of clotting. Last thing are the white blood cell types and the normal differentiation. The white blood cells is about 4 to 11,000 per cubic milliliters. We can describe it by percentage and by absolute number. The white blood cells are composed of two important kinds, those having granules inside them and we call it granulocytes, and those without granules we call it agranulocytes. For the granulocytes we have three forms, one called eosinophils and one called basophils, and the neutrophil, which is composed of non-eosinophil and non-basophils. The lymphocytes and the monocytes are kinds that doesn't have granules or having very little amount of granules. These are the pictures of the neutrophils and basophils. The granulocytes also have another name, which is called the polymorph nuclear, because it has not a single nucleus, have more than one nucleus, and they have many different shapes of this nuclei. That's why they are called polymorph nuclears, because they have difference, and the granulocytes because they have granules. The neutrophils compose about 60 to 70%, lymphocytes about 20 to 40%, the monocytes is a scarce 2 to 8%, eosinophils about 1 to 4%, and basophils is very scarce, less than 1%, because it releases histamine and it is responsible for allergic response and defending against some of the parasites. So the most abundant is the neutrophils, which attacks the bacteria. So will be part 2. We will discuss the blood investigations, the common kind of blood investigations that we will use, and how to go through these investigations by simple scenarios of some of the important diseases. So keep on and see the next lecture, inshallah. Assalamu alaikum wa rahmatullah.